Hey everybody, I'm Bill.
I'm Tony, and we're both senior CNCs studying computer science.
So our project basically involves predicting membrane localization and protein expression for channelrhodopsins via various ML techniques. Channelrhodopsins are a type of light-gated ion channel, which are very useful in analyzing neuronal circuitry. A key engineering challenge in the design of novel channelrhodopsins is ensuring that they're able to properly localize to the plasma membrane, while ensuring that other properties that we desire are kept. A common technique used in protein engineering is a method called directed evolution, in which given a protein of interest, say a channelrhodopsin, we figure out the corresponding nucleotide sequence that encodes it. We perform random mutagenesis on this protein of interest. We then screen it to figure out which proteins are the highest performant. We then select the highest performing protein and perform another round of random mutagenesis on the amino acid sequence that gives us another library variance, and so on and so forth. This will ultimately give us a protein that is optimized for whatever task we desire. However, the key bottleneck is that this requires a lot of in-lab experimentation, which can be expensive and time-consuming. A paradigm shift is basically using ML techniques to tackle the underlying problem of figuring out which sequences are likely to be the highest performing, or which sequences encode the highest performing proteins. And this is a technique called machine learning-based directed evolution. And that's the overall goal of our project, in which we're building sequence to functional fitness models via various ML techniques. And we have a dataset that does exactly this. Our dataset consists of 218 protein variants, in which each variant has nucleotide data of around, say, 1,000 or so nucleotides, as well as the corresponding fitness metrics, for instance, protein expression and MK and membrane localization, which are both represented as scalars. If you take a look at the figure below, this represents the distribution of our output variables, in which you can see certain things like GFP mean, which represents membrane localization. For most of our samples, most of the proteins do not localize very well, and the same for MK mean, which is our metric for protein expression. For our testing, we basically did a bunch of different types of feature representations. For instance, given our nucleotide sequence data, say of length k, as a baseline, we used one-hot encoding, in which essentially our sequence of length k is transformed into a matrix of size k by 6, k being the number of nucleotides in our sequence, and 6 because our alphabet contains 6 different characters, the 4 nucleotides, and a start of sequence token and an end of sequence token. We fed these one-hot encoding matrices into either a flattened version into an MLP, or we fed it directly into an LSTM. And we hypothesized that LSTM would perform better because LSTMs, in principle, work well in sequential data. As another representation, we considered something called the KS-game representation, which is an image-like embedding of a string that displays local and global patterns. If you take a look at the figure below, given some sequence like ATCT, we feed this into the CGR algorithm with a parameter called the Kamer size, which in this case is 5. And this results in a 2D plot shown on the right here, which basically encodes local and global patterns, and we feed this image into a suite of different CNNs.
Another input feature technique we have experimented is using DNA-BERT pre-trained embeddings. And DNA-BERT is an analog for BERT in NLP, except that instead of inputting a sentence, we now have a nucleotide sequence as input. So for each of the nucleotide, A, T, C, and G is going to be transformed into a 768 dimensional embedding. We're able to download the model from an open source called Hugging Face and fine-tune the pre-trained embeddings onto the multi-layer perceptrons and LSTMs. This page is our results, where the blue lines represent the training losses and orange lines represent the validation losses. The top row are the results from our baseline models. As you can see, the validation losses tend to jump up and down quite a bit. This is because our data is quite noisy, and we only have 218 data points in total. You can also see that the training losses are consistently lower than the validation losses, indicating that there is a certain degree of overfitting for the baseline models. The bottom row represents the performance of our CGR-based models. As you can see, for these models, the training and validation losses are much more stable, and we're able to achieve the best performing model with a CNN and a CGR representation, and we're able to achieve a R-squared of 0.32 on the test dataset.
discussion, we hypothesized that the reason why our CGR-based models were able to perform better than other types of feature embeddings is simply because CGR is more compact and has literally a smaller embedding. For instance, for a camera size of 5, this results in, regardless of the size of the input, this results in a 32 by 32 image. Whereas for our one-hot encoding, this requires a matrix of size around 7,000, which is 7 times bigger than the CGR representation. Furthermore, as for some next steps, we can consider converting the nucleotide codons into amino acids and running various analogs for our DNA-based models on our amino acid models. For instance, protein BERT is an analog to DNA BERT, and 3D CGR is an analog to normal CGR for proteins. We hypothesize that these methods may be more compact and may have better model performances. Thank you.
Thank you.